In this podcast, you are going to listen about WBC practical viva questions with answers. Let us start. First question: which diluting fluid do you use to calculate WBC? The name of the fluid, that is Turk's fluid. Next question: what is the composition and functions of Turk's fluid? Turk's fluid composition: glacial acetic acid, junction violet, and distilled water. Glacial acetic acid 3 ml, it destroys the membranes of RBCs, platelet, and imparts refractarity to the cells. Junction violet about 1 ml, they stains the nuclei of the WBC. Distilled water acts as a solvent. Next question: how will you identify the pipette, WBC pipette? WBC pipette can be identified by observing the color of the mouthpiece, size of the bulb, and presence of bead in the bulb and markings on the pipette. In WBC pipette, the markings are 0.5, 1 and 11. In case of RBC pipette, 0.5, 1 and 101. Next question: what are the units on the WBC pipette? On WBC pipette and also on the RBC pipette, the markings has no units. We just call them as parts. Next question: tell the procedure for WBC count. We have to sterilize our finger with a spirited gauge, give a deep prick, discard the first drop of the blood, and take the blood up to the 0.5 mark. Next, take the WBC diluting fluid up to the 11 mark. Wait for 3 to 4 minutes. Discard the first 2 to 3 drops of the solution and go for the charging. Next question: how many squares do you count for WBC? Total 4 large corner squares we will use for the counting. Those are further divided into 4 into 4, 16 squares. Next question: which objective lens do you use for WBC counting and how you will adjust? 10x objective lens we will use for the counting of the WBC. The adjustments are: condenser is in lowest position, iris diaphragm used to be closed or slightly opened. Next question: what is the formula for dilution factor? The formula for the dilution factor: the final volume achieved by the original volume of blood has taken. The dilution factor in WBC count is 1:20. Next question: what is the final formula for WBC count? N into 50 is the final formula for WBC count. Next question: why we should discard first 2 to 3 drops of a solution before the charging? Before the charging, we have to discard the first 2 to 3 drops because the fluid present in the stem portion is not containing any blood. So that portion is free of cells. We have to discard before the charging. Next question: which method do you use for the counting? For the counting, just like in RBC, we can use either L method or inverted L method to avoid false positive or false negative results. Next question: what is meant by glacial acetic acid? The term glacial means pure acetic acid. Only the glacial acetic acid can give the typical shine or clear refractality around the WBC due to swelling of nuclei. This differentiates them from the dust particle, which are opaque and of different shapes. It is called glacial because during its manufacture, it gives the appearance of a glacier at a one stage. Next question. How long an ideally charged chamber preparation will be lost? Usually it lasts for 90 minutes. However, glacial acidic is unstable, it's a volatile, so it's a stable up to 40 minutes only. So it is better to complete the practical within 30 minutes. Otherwise, it will dry up. In the dried area, we cannot absorb the cells. Next question. Do glacial acetic acid affects the membranes of the WBC? No. The glacial acetic acid, the concentration that we are using in this solution, it is only affects the membranes of the RBCs and platelets. It is not enough strength to break the membranes of the WBC. Next question. Why the RBC not seen in WBC counting? RBCs are not seen in WBC counting because those membranes, RBC membranes, were destroyed by the glacial acetic acid. We can see the debris of the RBC, not the cell. Next question. Can any other agent be used to immobilize the RBC? Answer is no, because any weak hemolytic agent will take lot of time to lyse the RBCs. On the other hand, any strong agent will lyse the WBC nuclear membrane also. So that's why we should not use any other acid. Next question. What are the source of error in WBC counting? The source of error, pipette error, chamber error and field error may occur. Next question. What is the difference between total leukocyte count and differential leukocyte count? Answer. In total leukocyte count, we used to calculate all the numbers of a WBC in absolute value. In case of differential leukocyte count, we used to count the cell in percentages per 100 cells. Next question. What is the normal count of WBC? It is 4,000 to 11,000 cells per millimeter cube of blood. Next question. What is leukocytosis? And write the physiological and pathological variations. Leukocytosis means increasing number of WBC. Both physiological conditions and pathological conditions we can observe the leukocytosis. Physiological leukocytosis we can observe in normal infants. It is a high. After taking the food, we call it as a digestive leukocytosis. After physical exercise, during the mental stress, in the pregnancy, during the maturation, and exposure to extremes of a temperature, we can observe rise of total number of leukocyte count. Pathological variations. Leukocytosis occurs in acute infections with a pyogenic or pus-forming bacteria, myocardial infarctions, acute hemorrhage, burns, amoebic hepatitis, malignancies, and surgical operations. Next question. What is leukopenia? Decreased total leukocyte count number, we call it as a leukopenia, occurs in both physiological and pathological conditions. So, physiological leukopenia we cannot observe casually. It is unusual and rare. Next, coming to the pathological leukopenia. So we can observe in infections with non-pyogenic organisms like typhoid and paratyphoid fevers, and sometimes in protozoal infections like malaria, and viral infections like influenza, mumps, smallpox, AIDS, and drugs like chloramphenicol, sulfonamides, aspirin, penicillins, cyclosporins, phenytoin, etc. These cytotoxic drugs used in treating malignancies may also cause leukopenia by depressing the bone marrow. Next, repeated exposures to the X-rays and radium, chemical poisons that depress the bone marrow like arsenic, dinitrophenol, antimony, and others, and malnutrition and hypoplasia and aplasia. In these conditions, pathological leukopenia we used to be observed. Next question. What is the difference between leukocytosis and leukemia? Leukocytosis means increase in total leukocyte count above 11,000 cells per millimeter cube of blood, whereas leukemia is a cancerous condition of the blood in which total leukocyte count usually more than 50,000 per millimeter cube of blood. Next question. Classify the WBC. Classification of WBC based on the granules, granulocytes, and agranulocytes. Granulocytes are neutrophils, eosinophils, basophils. Agranulocytes were monocytes and lymphocytes. Next, what is the difference between leukocytosis, leukostasis, and leukemioid reaction and leukemia? Leukocytosis means simple increase in total leukocyte count number. Leukostasis means if the count is more than 1 lakh per millimeter cube of blood, we called white cell thrombi may form in the brain, lung, and heart. This condition we call it as a leukostasis. Leukemioid reaction. It is an extreme elevation of the total leukocyte count, that is more than 50,000 cells per millimeter cube of blood as a result of the presence of mature or immature neutrophils. Leukemia. It is a cancerous growth of the WBC cells more than 50,000 cells per millimeter cube of blood we can observe. Next question. What do you know about bone marrow transplantation and what are its indications? Bone marrow transplantation is the transplantation of healthy bone marrow from the donor to the recipient. The purpose is to establish normal hemopoiesis, and so normal blood cell count is in the recipient. The procedure has been successfully used to treat certain types of leukemias, some cancers, severe combined immune deficiency diseases, some genetic disorders, and hemolytic anemias. In this, the patient's defective red bone marrow must be destroyed fast by whole body irradiation and a high dose of chemotherapy. Then the donor's marrow must of course be closely matched with that of the recipient to avoid rejection. The stem cells from the donor's marrow settle down and start to grow. Thank you for listening and stay tuned for the further updates. This may help you for your exam attention. Thank you.